Thanks for joining us on another exciting journey into the world of science, right here on the Sanson Science Experience. If you enjoy this kind of content, hit like and subscribe. It's the best way to support the channel and keep these episodes coming. All right, let's jump into today's deep dive. We're tackling a really tough medical problem, severe mitochondrial diseases, the kind that particularly wreck the cell's energy production.
Yeah, they can lead to devastating conditions like encephalopathy, especially in tissues that use a ton of energy, like, well, the brain.
Exactly, and these are genetic disorders, right? Glitches in the DNA.
That's right, either in the mitochondrial DNA itself or in the nucleo-DNA that codes for mitochondrial parts. And the really tough part, there aren't really any effective treatments right now that, fix the underlying cause.
It's mostly just supportive care.
Mostly supportive care, yeah. Which is why this new research is so potentially exciting.
Yeah, we found this fascinating article looking at a completely different angle of potential gene therapy. They tested it out in mouse models.
So our mission today is to unpack this research. We want to understand their approach, look at the results, and really dig into their findings.
Because they found something pretty surprising about restoring energy function in neurons, didn't they?
They really did. It kind of challenges some existing ideas.
Okay, let's set the stage. Before we dive into the therapy itself, maybe we should quickly recap the system they're trying to fix. OXPHOS.
Good idea. So OXPHOS, or oxidative phosphorylation, it's basically the main power plant in our cells. It's where most of the energy currency ATP gets made.
And it involves a whole chain of protein complexes.
Exactly. 5 big complexes embedded in the mitochondrial membrane. This study focuses specifically on problems with complex 1 and complex 4.
Complex 1 being the big entry point, and complex 4, that's cytochrome C oxidase.
That's the one, a crucial final step in the chain.
So if the genetic instructions for building these are faulty, the power plant essentially grinds to a halt.
And for tissues like the brain that need constant energy, that's, catastrophic.
Right. Complex 1 defects, for instance, can cause things like Lee syndrome, ataxia.
Yeah, serious movement and coordination problems, developmental delays. It's often very severe. Complex 1 issues are actually a major cause of these mitochondrial diseases.
And complex 4 defects are just as bad. I see hypertrophic cardiomyopathy listed here, more ataxia, lactic acid buildup.
Absolutely. And Lee syndrome again, it just hammers home how desperately we need better therapies than just supportive care.
Which brings us back to the gene therapy idea.
Right. The core concept is pretty straightforward. If a gene is broken, can we deliver a working copy to fix the problem?
And they used a virus to do that, an AAV.
Yes, specifically a type called adeno-associated virus, or AAV. So they used a particular variant, AAV-PHP.eB.
Why that one?
Well, in the specific mouse strain they used, this variant is known to be quite good at getting across the blood-brain barrier and delivering its genetic payload into the central nervous system, the brain and spinal cord.
And the goal was to get it specifically into the neurons, the brain cells themselves.
Precisely. To deliver a working copy of either NDUFS3, which is needed for complex 1, or COX-10, which is essential for complex 4, depending on the disease model.
And how do they model the disease in the mice?
They engineered them very cleverly. They created what are called neuron-specific knockouts.
Meaning the faulty gene was only switched off in the neurons.
Exactly. They used a genetic tool called CAM-KIA-CRE to do that. This way, the mice develop progressive neurological problems that really mimic what happens in human patients, where the brain cells are the primary issue.
So they had these mice born with a defect just in their neurons, kind of like the human condition.
Right. Then they gave them the gene therapy.
Oh, a single shot?
Yep, a single retro-orbital injection that's near the eye when the mice were young, before symptoms really started showing. It's known to be efficient for brain delivery in this particular mouse line.
And critically, they needed a control group.
Absolutely. They had similar knockout mice that got a control virus instead. This one carried a harmless marker gene, EGFP green fluorescent protein, not the therapeutic one. So that group showed what happens without the treatment.
Okay, let's get to the results.
Yeah?
Because this is where things sound pretty dramatic. Behaviorally.
Oh, it was night and day. The untreated mice, the controls, they went downhill as expected.
Short lifespan.
Yeah, dead by 5 to 10 months usually. They lost weight, developed that hunched posture you see in sick mice.
And problems moving.
Definitely. On tests like the Rotarod, which measures balance and coordination, or just observing them in an open field, they were clearly impaired. major motor deficits.
And the mice that got the gene therapy.
It was an incredible rescue. Their survival was massively extended, living beyond 15 months, basically a normal mouse lifespan.
Wow.
They kept their weight up, looked normal, moved normally. On those same motor tests, they performed just like healthy, wild-type mice. It wasn't just a small improvement. It looked like a complete prevention of the behavioral disease.
That's amazing on the outside. But did they actually fix the problem inside the brain cells, the energy production, or was it just masking things?
That's the crucial question, right? And the molecular data really sealed the deal. It showed the therapy was working at the fundamental level.
How so?
Well, first they looked at the proteins. In the complex 1 mice, they saw that the levels of the missing NDUFS3 protein were restored in key brain areas, like the cortex and hippocampus. And another protein, NDUFB8, that depends on NDUFS3, also came back up.
Okay, so the missing part was put back. What about the complex 4 model?
Same story. They delivered the COX-10 gene, and they saw restored levels of the COX-1 and COX-4 proteins, which need COX-10 to be properly assembled into the final complex 4. So the building blocks were definitely being made again.
But were they assembling correctly into functional power units?
Yes. They used a technique called BNPG, which lets you see the big intact protein complexes, and it showed that proper complex 1 and complex 4 structures were forming again in the treated mice brains.
Okay, structure restored. What about function? Were they actually working?
They check that too, using in-gel activity assays. Basically, they run the complexes on a gel and then add the chemicals they normally process to see if the reaction happens. And it did. They saw restored enzymatic activity for both complex 1 and complex 4. The power plants weren't just rebuilt, they were switched back on and generating energy.
Incredible. So, function restored. What about the damage control? Did it stop the neuroinflammation and cell death?
Largely, yes. They looked at markers for brain inflammation. GFAP, which indicates reactive astrocytes, support cells that get activated during injury, and IBA1, a marker for activated microglia, the brain's immune cells. Both of these were significantly reduced in the treated mice compared to the controls.
And this reduction in inflammation correlated with less damage.
Exactly. They saw much less neuronal loss and prevention of the decrease in brain mass that you see in the untreated knockout mice. The treated brains looked much healthier.
So less inflammation, fewer dying neurons.
Right. Although it's interesting, they noted that even in the older treated mice, there were still some mild signs of inflammation lingering in certain areas. Not totally gone, but vastly improved.
Okay, so far this sounds almost too good to be true. Healthy mice, restored energy function, reduced brain damage, a home run for gene therapy, right?
You'd think so. It's incredibly promising. But here's the kicker, the really surprising part of this study, the twist.
Uh-oh. What is it?
Despite this amazing rescue across the board, the gene therapy didn't actually get into every neuron. Not even close.
Wait, what? You mean not all the target cells got the fix?
Nowhere near all of them.
Yeah.
They figured this out by looking at those control mice that got the virus carrying the GFP gene instead of the therapeutic one.
The green fluorescent protein.
Yeah. Since it's delivered by the same AAV vector, where you see green tells you which cells actually took up the virus, they stained brain slices for GFP and for NUN, a marker for neurons, and then they counted.
And what percentage of neurons actually got the gene?
It was only about 23% to 30% in the brain regions. They looked at motor cortex, piriform cortex, hippocampus.
Hold on. Only 30%?
Yeah.
Roughly.
Yeah.
You're saying fixing less than a third of the neurons was enough to completely rescue the mice from this fatal disease?
That's exactly what they found. That's the core counterintuitive conclusion. You get these massive life-saving benefits, even though the majority of neurons didn't receive the gene therapy directly.
That just, wow. It implies the neurons that weren't fixed were somehow being helped out.
It has to mean that, right? They must have been getting support or protection from their local environment, including those neurons that were corrected.
That changes how you think about needing to fix every single cell.
It really does. And they actually had another piece of data that sort of supports this idea of a network effect, or maybe a threshold.
What was that?
They used a different, less efficient genetic system, a weaker Cree, to create the knockout. This actually resulted in more neurons having the defect, maybe over 50% they estimated. But interestingly, these mice didn't develop the severe disease phenotype nearly as quickly, at least not within the first 6 months they observed.
So even with half the neurons being faulty, the system could still cope reasonably well.
It suggests there might be a certain threshold of overall OPS-PHOS function needed in the network. If you stay above that threshold, maybe the brain is more resilient. And that could explain why fixing just 30% with gene therapy works so well. You push the whole network back over that critical functional threshold. Or maybe the corrected cells provide enough support to the others.
Okay, so the million-dollar question is how? How does fixing only some cells rescue the whole system so effectively? What are the possible mechanisms?
The researchers proposed a couple of really interesting ideas. One is mitochondrial transfer.
Cells sharing mitochondria, like passing them around.
Yeah, exactly. There's growing evidence that cells, including neurons and maybe the supporting glial cells, can actually transfer healthy mitochondria between each other. So it's possible the corrected neurons were literally donating working power packs to their struggling neighbors.
Like sending over spare batteries. That's wild.
It is pretty wild, but it's a known biological phenomenon. Another possibility is more about the overall environment.
How so?
The gene therapy dramatically reduced neuroinflammation, right? Maybe just cleaning up that toxic, inflamed environment created by the disease was enough to help the uncorrected neurons survive and function better.
So less stress, a healthier neighborhood helps everyone, even the ones who didn't get the direct fix.
Precisely. A less hostile, more neuroprotective milieu could make a huge difference to those neurons still limping along with faulty mitochondria.
So, thinking about taking this to people, what does this partial correction finding mean?
It's potentially huge for translation. It suggests that for these devastating mitochondrial brain diseases, a gene therapy might not need to achieve 100% correction in every single affected neuron to provide a really significant, maybe even life-saving clinical benefit.
That definitely lowers the technical bar, which has always been a major challenge for brain gene therapy.
It's certainly encouraging,
Sure.
but, we have to temper that enthusiasm with the real-world challenges that still exist.
Right. What are some of the big hurdles they mentioned?
Well, a major one is the vector itself. This AAV-PHP.eB worked great in these specific mice, but its efficiency doesn't automatically translate to larger animals like primates or to humans. We need vectors that work effectively and safely in people.
That's a common issue in gene therapy development, isn't it?
It is. Another big consideration is timing. Many of these mitochondrial diseases hit really hard, really early in life.
Like in babies or young children.
Exactly. For the most severe forms, the ideal time to intervene might be incredibly early. People are even talking about the possibility of fetal genetic testing and potentially treating in utero before birth. That opens up a whole different level of complexity technically and ethically.
Wow. And then there's just the practical side of dosing the virus.
Yeah, finding that sweet spot. You need enough virus to get sufficient correction, but not so much that it causes toxicity or triggers a strong immune reaction against the vector itself. It's a delicate balance.
And figuring out who would benefit most. Is there a point where the disease is too advanced?
That's another key question. Is there a biochemical point of no return, a stage where the brain damage is too widespread for even this kind of partial rescue to work effectively? Defining that threshold will be crucial for deciding which patients might benefit and when to treat them.
Okay, so let's wrap up this deep dive. We've looked at this really promising gene therapy approach for these awful mitochondrial brain diseases in mice.
And the results were genuinely remarkable. Mice living normal lifespans, behaving normally, with clear evidence that the energy production was fixed at the molecular level and brain damage was significantly reduced.
But the real mind-bender, the take-home message that's got us thinking, is that all this was achieved by fixing only a fraction, maybe around 30% of the neurons.
Yeah, that partial correction yielding such a full benefit is the truly surprising and honestly hopeful finding here.
It really makes you reconsider how interconnected the brain is, doesn't it?
Absolutely. And it raises fascinating questions. Could this kind of partial rescue strategy be applicable to other neurological disorders too? Could boosting the function of a subset of cells be enough to support the wider network in conditions beyond mitochondrial disease?
What amazing possibilities does that open up for future research into brain function and repair? Lots to think about. Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already.